Hello, welcome everyone to the online series of videos lectures by Rama University. Myself, Dr. Simranjit Singh from the Department of Biotechnology, and today I will be taking to the world of proteomics. So at the starting in this video lectures, I will be mainly taking care of the world of proteomics, the basics, that what is the basics proteomics means and how it could be beneficial in our day-to-day life. So this will be quite beneficial for all the undergraduate or postgraduate students who wants to study about proteomics, because some of you might be studying biotechnology, and sometimes there are subjects coming like proteomics. For them to understand the core of proteomics, which is quite important, will be quite difficult to understand the further advanced things in it. And from my background, basically I did my BTech in biotechnology from Dept Singh College in Punjab, Chandigarh. And then my thirst for knowledge didn't stop there. I went for my master's in agrobiotechnology from Germany in Gießen. And after finishing my master's there, I went for my PhD in heart failure field in the cardiovascular physiology, which I just finished in 2017 from University of Göttingen, also from Germany. And then later on, I switched to India, where I started as my career assistant professor at the Mata Gujari College in Fatehgarh Sahib. Now, currently, I've been assisted at the Rama University, where I'm teaching various subjects to the bachelors or the postgraduates. Variety of subjects related to biotechnology. So today I choose this topic because that was my actually core PhD topic, where I was studying about a redox regulation of phosphatases in heart failure. And to do so, my core study was related to the the thiol modifications, the how thiol is working out, how things are working at that side. But we will not go in that way in today's lecture. We will be more focused about very basics about proteomics. What is proteomics? What are its various disciplinary branches are? What is the basic foundations of proteomics, and how it could be, make your base before we go in advance. So let's start on that note. I think I should go like this so that it could. So this is my first time I'm giving this lecture, so excuse me if somewhere I goes wrong. Yeah. So today's lecture is about world of proteomics, where we will be understanding the basic definition. What is proteomics is all about? Basically, it's nothing. It's just to study about all the proteins in our body. If you have remember, that is a central dogma of life, which means that from DNA, your mRNA is made, from mRNA, your protein is made. From DNA to mRNA, the process is called as transcription. And from mRNA to production of your protein, this process is known as translation. And this whole process of DNA to mRNA to proteins together makes your central dogma of life. And we are in particular at the moment not interested in the DNA, not interested in the mRNA, but interested in the end product, that is your protein being produced in the whole chain of your cell. So before you might be understanding these topics, I might think that you are might aware of core cell biology, molecular biology. So you might be aware of these things. Only then you are able to understand. I hope you all know about that. If you doesn't know, please comment on the box. We will reply to that. So the basic definitions, it says that analysis of complete complements of proteins, identifications, you have to identify them, then quantify them, then modify them, then how they're interacting and how the activities is working out. So making this jumble, first you have to identify that how much they are present, the quantity is very important, how they are modified later on. This is also quite essential. Then how they're interacting between each other and making their activities. Quite important in these things. So that's the basic definition. And beyond that, another definition we can say is that it's a analysis of protein complements to the genome. So if, as you have discussed, that from gene, after the transcription, your protein is produced. The study of gene here is known to be as genomics, where the study of protein is known as to be proteomics. But if we compare from genomics to proteomics, because in our body, we have lots of proteins, lots of proteins we can't even think of. And like essentially there are 20 amino acids, of course, that will be discussed in this part now, but together they make lots of proteins in our body, and to study their interaction and to study their modifications, to study their post-translational modifications, to study their basic activities, how they're working, it's much complex than your genomics part here. So remember that. So proteomics is much complex than your genomics part here. And beyond that, you can see is how proteomics is interlinked with the other branches. So there is expression of your protein. If you have remember, there is a thing called 2D electrophoresis or SDS page, you know. So these are the things that we do it for 2D electrophoresis or SDS page. So these techniques, we do it to check your protein quantification, like which protein is present, which protein is present in your sample. So to do that, we do the expression, and to do the 3D structure, to check 3D structure, we do the X-ray crystallography. Sorry for my writing, but I hope it is visible to you. So in the X-ray crystallography, it means that you take your sample of your protein that you want to look for, you are not aware of. You make a crystal of it, then you put X-ray light on it, you diffract the X-ray diffractions, then analyze on the computer, and then whatever the structure is being predicted with the help of softwares, you can say that that protein is hemoglobin, that protein is arginine, or that protein is phosphatases or kinases or oxidases, any type of protein it could be. Now let's check further. So these expressions of proteins, 3D structures, they are interlinked with their post-translational modifications. So in these proteins, there could be some changes could happen, like disulfide formation could happen, you know, acetylation could happen, methylation could happen, hemolydation could happen. So all these things are possible at the post-translational level. Like after the translation, whatever the changes are happening, that is known as your post-translational modification. Yeah. So together, your expression, PTM, 3D structure, they correspond to your proteomics. And to study, we can study all these things for the interactions, how all these proteins are interlinked with each other and interacting for the proper functioning of body. So if you remember, if somebody is doing some gymming or going to gym or doing some exercise, so all the gym athletes, they are taking whey proteins from outside, and they're also taking keratin, BCCA, all these supplements also for the building of their muscles and better metabolism. So all these things, if you want to increase your muscles, of course, you have to increase your body mass, and to do so, you cannot suddenly like increase it with the normal eating food. Yeah. So normally the bodybuilders from the villagers, so they're eating desi ghee, and that is your ghee oil made from cow milk, and that is quite essential for building of the muscles. But nowadays it has been transformed into the artificial market with all whey proteins in the market. So they are also quite helping you to build muscles, and you can see some changes in your body within few weeks. So the changes that you might see in months, 3 months, but you can see within 2 weeks, you can gain your muscles and things could be developed. And this is all happening because of proteins. And beyond that, all these studies could not be stopped at this level. So you can also take the help of bioinformatics, like all the softwares that could help you to understand these interactions at the computational level, at the computational biology level. So all these interactions, they are quite important. And beyond this, the proteomics is quite essential. It's a multidisciplinary field which could be used from the biology, bioinformatics, molecular biology, protein biochemistry, analytical chemistry. So all these things are all involved in your studies. Yeah. Then further, yeah, what are the applications of proteomics? So basically, proteomics helps you to study any disformation is happening in your body, or any good changes are happening in your body. So let's say, for my example, my sister is autistic by childhood. We were not aware till she was 4 years old, and my father and mother doesn't know about it, but she started to show some autistic characteristics in her day-to-day life. So we went for genetic analysis, and we found that initially that her brain from the left to right, they were not normal, and beyond that there were some genetical mutations that happened in her body, which is not normal in the human body. So these are all also happening at the protein level, mutational level, at the post-translational level, and that's why we are seeing these things. So still scientists are finding which protein was the reason for that, why we are seeing these diseases sometimes, and why we cannot cure with normal medicines, because there's no medicines for autism also till now. And same way, some other applications of proteomics here we can see is like hemoglobin, which picks up your oxygen in the lungs and travels through blood and delivers to the cells. So here you can see that oxygen is transferring to your red blood cells, and it's transferring from one region to another. So it's helping to your proper functioning of your oxygen in your body. Then if there is single amino acid change is happening, so what happened, let's say these are the various normal human globin with M, V, H, L, T, P, E, E. So this is sickle cell. So this is normal round cells of your red blood cells. But what will happen, instead of E here, your valine is present here. So this is mutated one. So your shape of your the normal shape will be now sickle cell. So this leads to your sickle cell anemia. So there will be not proper functioning of your blood, and it will not carry the same amount of oxygen as it was carrying in the circular form. So there will be always a depletion of oxygen in the body because of that. So this is also happening at the post-translational level, and we are able to understand this because of your proteomics. So why? So on another aspect, that how proteomics changes from of our body, from a baby that is born just 2 months old, and being a 33 years old man, how the proteome of that person will be changed. Same hold for if there is a caterpillar, butterfly egg is there, then it's becoming a caterpillar, then there's a chrysalis being attached, pupa, then it's become an adult butterfly, then it's become again egg. Then from egg to caterpillar to chrysalis to adult butterfly, in all these different stages, the proteomics of these various organisms will be different. It will be different at different stages. It will be modified. It will become bigger. It will get more complex. It will make more advances in the later ages, and it will start to deteriorate also at the later ages. So that's why at the later ages in the human beings, it's in the Western country, not in India at the moment, Alzheimer's disease is quite being prominent at the moment, where persons start to dissociate their their disease, they cannot think, their memory is lost, and it's quite prominent there, and and still scientists are not able to find what could be the reason behind it. So this is, of course, some changes are happening at the post-translational level. Something is not being done at the better level. So that's why these things are happening. So one organism will radically different protein expressions in different parts of body. So your proteins will be different in different parts of body, and in different stages of life and different environmental conditions. So it will be different at the different stages. That was one aspect. So I will not take you much time. So already taken your 15 minutes here. So let me take you to the other part. So why should we study proteins actually? What is the reason behind it? What will gain from it? So far we have understood that it has quite important functions in the proteins, in the disease, in the day-to-day life, from the making our body to proper functioning. But why should we study it? Because it has lots of function in our body. It has many advantages, as we can see, like motion. Like for the motion of our body, the actin muscle compartments, it contract muscles. Beyond that, enzymes. So various enzymes in our body, like proteases, or which degrades proteins, they are quite essential also. Then the regulations like insulins, which controls blood glucose in our body. Then defense systems to fight diseases with the help of antibodies. It also helps to produce hair and nails. So our nails are also proteins actually. Hairs also being produced are form of proteins itself, known as keratin. And also, as we discussed, as a hemoglobin which carries oxygen is also in the team of proteins here. So as in all, it is quite essential. Without proteins we cannot exist, and our life would be really disaster. So that's why I choose this topic for you today, to make you comfortable with this topic, to make you excited actually. So my reason main here is not to just give you this lecture, but to ignite a fire of knowledge that you want to learn more about proteomics, and you can go further into your studies for your better understanding and sharp knowledge regarding the same. So now I will take you to the basics part of proteomics. So what is how protein is made up of, what are the basic structures. So you might have studied in your class 11th or 12th. Those students who are from class 11, 12, it will be quite essential for them. Those who have not studied, let's learn from it. So proteins are composed of amino acids. Here we can see there is one variable group, that is R. Because of this, you will have different amino acids properties, and then you have your amino acids group, that is NH, which act as a base, and there is a carboxylic group which act as an acid. So that's the basic component of your amino acid. Yeah, so that's the basic. Then what will happen? One of your amino acids here, amino acid 1, amino acid 2, so they will come and they will join together by removing this extra water. So from the carboxyl group, OH will be taken, and from the amine group, H will be taken. So we will remove this one, and together they will form a peptide bond. And this peptide together, this structure will be called as dipeptide bond. Yeah, this together, the structure will be called as dipeptide bond. And in this way, you have one carboxyl group, one amino acid, second, 2, 3, then amine group, 1, 2, 3, like this. They will make a short chain of amino acids, and they will be called as peptides. And together, these proteins of polypeptides, they contain many peptides. This is known as your polypeptide. And together, this is known as your protein. And this polypeptide could be made up of various different kinds of proteins, amino acids, sorry. And these could be like your basic, acidic, non-polar hydrophobic, and polar hydrophilic. So the basic one will have the more basic properties like histidine, lysine, arginine. I will not go to the more structure about them, because that will take whole one lecture about it, how the structure is made up of. And it's all because of the actually this variable group in it. So it's histidine, lysine, arginine. Then there is acidic, that is aspartate, glutamate. Then there is a non-polar hydrophobic, that is alanine, valine, leucine, isoleucine, proline, and many more. And then in the polar hydrophilic, glycine, serine, glutamine, tyrosine, some few examples, and some more into that actually. So as you can see in this whole picture, I will remove myself from it for some time. You can take some screenshots from this video. So what you can study, what you have studied today, I will make a summary. You have studied the basics definition of proteomics, how proteomics is quite complicated. We have understood the multiple disciplinary aspects of proteomics. And then we went for basics applications of proteomics, how they are helpful in our day-to-day life, and why should we study these proteins. Then we went for the basics structure of amino acids, how they're made up of. We study about how they come together, they make polypeptides, and what are the various types of amino acids. And furthermore, we have studied how at the different stages of our life, this protein of our body, proteomics, is being gradually changing. So on that note, I will say thank you everyone for being a nice lecture listener to me. And so it's almost 20 minutes, so I have given on a right time. And if you have any questions, any comments, please write them in the comment box. We will reply to them. And beyond that, from my point of view, keep curious, like Steve Jobs says. Keep learning, keep be foolish that you don't know anything and you want to learn more. Yeah, that's why we are here for. And life is all about learning, and we should not stop learning. Thank you very much. Sat Shri Akal.